The first step in the process of protein synthesis is called transcription. Transcription cannot begin until it is initiated in the promoter, a region in the DNA. RNA polymerase and proteins called transcription factors attach to the promoter, causing transcription to begin. In transcription, the enzyme RNA polymerase II unwinds the DNA in the nucleus of the cell. Transcription is the conversion of the gene code of the DNA into a strand that is complementary in RNA. This is done by copying the DNA strand. The DNA strand, or the template strand, gives the growing strand, or the RNA, the pattern of bases to use as complements. Guanine matches up with cytosine, and adenine matches up with thymine, but is replaced by uracil in RNA. When these nitrogen bases are added, they are attached to the 3' end of the RNA.
The RNA coding sequence of the gene is transcribed to make pre-mRNA, which contains both introns and exons. Exon shuffling occurs when creating mRNA and only occurs in eukaryotic cells. Introns are interrupting sequences of nucleotides that do not code for the protein. Exons are DNA sequences that code for and express the protein. In eukaryotes, only RNA processing removes the introns and recombines exons to form mRNA. The mRNA is made up of a leader, reading frame, and trailer. The leader and trailer are not translated, but are caps on the reading frame that also protect it. The reading frame is made up of codons that are made of 3 nucleotides that will match with the tRNA. After transcription, the mRNA leaves the nucleus to continue to translation in the cytoplasm. In the cytoplasm, there are tRNA that is made of a single strand of nucleotides that fold to form its unit shape with 3 distinctive loops. These 3 loops are formed by hydrogen bonds between their complementary nitrogen base pairs. On loop 2, the antigen bond that pairs with the new normal mRNA is exposed. An amino acid that then corresponds to the anticodon bonds to the 3' end of the tRNA. rRNA is also located in the cytoplasm, but it is made in the nucleus. rRNA makes up ribosomes, which have 1 small and 1 large subunit. Ribosomes have 3 binding sites, E, P, and A, for joining tRNA and rRNA. The start codon of mRNA enters the P site and is joined with the corresponding tRNA anticodon. The next codon sits in the A site, and tRNA anticodons match with the codon to make the amino acids. Amino acids at P and A sites are joined by a peptide bond. tRNA release their amino acids through the E site. By joining tRNA and rRNA, an amino acid sequence is made to build a protein.
Translation occurs in the cytoplasm and starts when mRNA binds to the small subunit of the rRNA. tRNA binds to the large subunit. In the ribosome, hydrogen bonds are formed between the complementary bases of the codons of mRNA and the anticodons of tRNA. Translation is initiated when tRNA with methionine and the anticodon UAG binds to the start codon of AUG on the mRNA. The initiator tRNA is in the P site of the rRNA, and the A site is available for the next tRNA bearing another amino acid. The tRNA in the A site binds with the next codon, and the adjacent amino acids found in P site and A site form a peptide bond. Then, the ribosome translocates the mRNA 1 codon to the right. Another tRNA enters the A site, and the tRNA that was in the P site is ejected when it reaches the E site. This process is continued until the stop codon is reached. The stop codon in the mRNA does not code for tRNA, but instead a release factor that allows for the polypeptide to be released. The rest of the tRNA is ejected, and the polypeptide chain is created. At this point, the mRNA is released along with the release factor in the polypeptide chain. More than one ribosome can translate on an mRNA at one time so that many more polypeptides can be simultaneously synthesized. Many ribosomes transcribing on one mRNA are called polysomes.